Hello, this is Marlene Gonzalez presenting the poster entitled Carbon Quantum Dots Synthesized using Sargassum SPP as a Fluorescent Bacterial Detector. As an introduction, every year, millions of people lose their lives due to the infectious disease caused by multi-drug-resistant bacteria. As a result, it is essential to have effective monitoring and combat methods in place in medical facilities, food, and environment. In this context, nanomaterials are presented as an alternative to solve this problem. Here, we propose using carbon quantum dots due to their low cytotoxicity, excellent biocompatibility, antimicrobial activity, and fluorescent properties in the specific imaging and detection of bacterial pathogens. Carbon quantum dots could be synthesized with various techniques. We used the hydrothermal method because it is cost-effective and eco-friendly. Carbon quantum dots were prepared by hydrothermal treatment of an aqueous extract of Sargassum SPP. In a typical synthesis, the extract was prepared by mixing 1 gram of Sargassum SPP with 50 milliliters of water using magnetic stirring at 60 Celsius degrees for 30 minutes. Then, the solution was transferred into a 100 milliliter Teflon lined autoclave and heated at 200 Celsius degrees for 4 hours. The resultant solution was dialysed against water for 48 hours. The product solution was dried under a vacuum and redispersed in distilled water at a concentration of 0.5 milligrams per milliliter for further characterization and use. The properties of the carbon quantum dots were evaluated through several characterization methods, such as scanning electron microscopy, dynamic light scattering, and zeta potential, to understand their particle morphology, hydrodynamic diameter, and surface charge, showing semispherical shapes, sizes around 5 nanometers, and high colloidal stability in aqueous media. Nevertheless, the more relevant properties for the application were their Fourier transform infrared and fluorescence emission spectra, which offer information on their chemical composition and spectral properties. Then, their capacity as fluorescent biomarkers to detect bacteria was analyzed using confocal microscopy. As can be seen, the fluorescence was highly influenced by concentration, figure 2a, and pH, figure 2b, suggesting surface state phenomena as a mechanism of fluorescence. This means that the fluorescence of the carbon quantum dots varies when their surface functional groups change. For example, they are quenched by acidifying the medium. Then, FTIR spectroscopy was employed to identify the functional groups, figure 2c, showing the presence of functional groups that are easy to protonate and deprotonate, allowing their use in sensing. In agreement with these results, the structure shown in figure 2d for the carbon quantum dots was suggested. The capability of carbon quantum dots to act as biomarkers was demonstrated using gram-positive Staphylococcus aureus and gram-negative Escherichia coli bacteria through confocal microscopy after an hour of interaction. Figure 3 shows that live Staphylococcus aureus bacteria emitted blue fluorescence, while dead ones emitted green light. However, the Escherichia coli strain showed no fluorescent signals under the same conditions. The ability of carbon quantum dots to selectively image gram-positive bacteria can be attributed to the difficulty of intracellular access of carbon quantum dots caused by the inner and outer lipid membranes of gram-negative bacteria. Additionally, the ability to distinguish between live and dead bacteria can be attributed to different interaction and permeabilizing membrane action between the carbon quantum dots and the bacteria. As conclusion, in this work, fluorescent carbon quantum dots were prepared by hydrothermal treatment using Sargassum SPP. The carbon quantum dots present semispherical shapes with diameters around 5 nanometers. Thanks to their structure, the carbon quantum dots present a tunable fluorescence that varies when their surface functional groups change. Therefore, When the carbon quantum dots interact with the membrane of the bacteria, they change the surface state, activating or deactivating different surface functional groups, causing a rate-shifted emission in the bacteria instead, allowing their use as bacterial detectors. Thank you for watching.